Okay, so the next person that I have a pleasure to introduce is Dr. Robert Hancock. Dr. Bob Hancock's current research interests include small catatonic peptides as novel antibiofilm agents and innate immune modulators, the development of early diagnostics for sepsis, and the systems biology of innate immunity, sepsis, and Pseudomonas, I've been practicing this word, aeruginosa. Thank you. He has published over 850 papers and reviews. He has 87 patents awarded to him. He is ranked third in microbiology worldwide with more than 149,000 citations and an H-index of 189. He has won numerous awards in Canada, 3 top prizes for health research, and is an officer of the Order of Canada. He is a co-founder of several companies, including ASAP Medical, Inc. Please help me welcome Dr. Bob Hancock.
Okay, I'd like to talk to you today about the diagnosis of severe sepsis and also endotypes at first clinical presentation. And you can see my conflict of interest statement. Both on the good side, which is the universities, and the dark side of the force, which is companies. So you'll have to take everything I say because I've decided if I'm going to discover something, I'm going to bring it to medical practice. Why is this not going forward? I'm pressing a green button. There we go. Okay, so sepsis is a life-threatening organ dysfunction caused by a dysregulated host response to infection. That is the official definition. Notice life-threatening organ dysfunction leads, and also the dysregulated host response to infection. And I think, quite frankly, infection is less important than actually what's going on in the host. So it's clinically diagnosed as suspected or documented infection. It can be either, and it usually is suspected, and an acute increase of 2 or more SOFA points, where SOFA is a sequential organ failure assessment dealing with 6 different systems in the body that presages organ dysfunction. You've already heard it causes 1 in 5 deaths, and I think this morning Alison put up a slide that said 1 in 4 deaths, maybe. And the COVID-19 pandemic is estimated to kill up to 18 million people, and virtually everybody who died from COVID died from viral sepsis. So viral sepsis is definitely a thing. And just for those of you who are thinking, well, we always have to use antibiotics for sepsis, in fact, you can only diagnose bacterial infection in about 50% of patients. So for the rest of them, we don't have a bacterial infection diagnosis. So viral sepsis may be a lot more common than we think. Another Canadian, Dr. Kumar, basically came up with the stat that there's a 7.6% increased of risk of death for every hour's delay in the diagnosis of septicemia. And most diagnostic methods have low accuracy in early sepsis. So this is the, in grey, the course of treatment in a patient. The patient presents to the emergency room with some kind of illness. The clinician, based on symptomology alone, will suspect sepsis and collect blood. And then there's an average of at least 13 hours. So if you start to think of that number, 7.6% increased risk of death for every hour, 13 hours is a very, very, very long time in that patient's medical journey. The symptoms are largely non-specific. They're things like altered heart rate, blood pressure, respiratory rate, etc. And therefore treatment is often delayed. And this results in poor patient outcomes, ICU admission where it costs a lot of money, and 22% on average deaths worldwide. There's a lot going on in these patients. We're not very good at dealing with this disease. So there are a whole bunch of different methods now that we try to diagnose sepsis. There are pathogen identification methods. There's the original concept of blood culture. There are now a whole bunch of multiplex PCR methodologies that sort out which actual organism you might have. And there's mass spectrometry of the pathogens in a person's blood. But remember, we only find an infection 50% of the time. There are also a whole bunch of biomarkers. You heard some of them in the last talk. Procalcitonin is probably the most prominent one, but none of these actually work very well, especially early on in a patient's medical journey. So in the emergency room, each of these markers has about a 50% accuracy, which means you could toss a coin up in the air and say sepsis or non-sepsis, and you'd be just as accurate as any of these tests. There are a whole bunch of machine learning algorithms that are now being produced that seek to collect data from all over the hospital about that particular patient, and the algorithm spits out to a risk of sepsis. These are more accurate, but there's a big logistic issue, which is, you'll know if you've tried to collect a lot of information in a hospital. It doesn't really happen in a very short period of time. And then most recently, there have been a bunch of people who are interested in measuring the host immune response. And there's this really nice test that was just improved from MeMed that distinguishes bacterial and viral infections with reasonable accuracy. And so I'm just going to tell you about one of these today. And the take-home from all of this stuff is that early markers and diagnosing infection have only approximately 50% success. About the same success as tossing a coin in the air and saying, heads your sepsis, tails you're not. Okay, so we were very interested in this particular phenomenon, and you can see in the graph over on the left-hand side, generally speaking, what's considered to be the inflammatory response in a sepsis patient. There's this initial event called the cytokine storm, where pro-inflammatory cytokines like tumor necrosis factor alpha are really elevated in these patients. And then basically after that, this inflammatory response dies down and goes to a hypoinflammatory phase where the inflammation is not only less than normal, but actually the patient no longer is able to respond to bacterial infections. And most of the deaths occur in this second phase rather than the primary phase, the cytokine storm phase. The major treatment for sepsis is antibiotics. There have been more than 30 clinical trials of testing anti-inflammatory agents, in other words, saying that cytokine storm is the most important thing going on in those patients, and none of them showed any benefit at all. It's considered the graveyard of biotech, not just because all the patients die, but because the therapies die in biotech, and so the companies die also. And we came up with this hypothesis that the hypoinflammatory phase was due to a phenomenon called endotoxin tolerance, which is that after one dose of endotoxin, a bacterial molecule, you get an inflammatory response. After 2 consecutive doses on human blood, for example, you actually suppress that response and you can no longer respond to bacterial infections. And the people who were involved in this are the individuals there. And the exceptionally handsome guy over on the left-hand side there happens to be my son, who did all of the early bioinformatics. So originally we were thinking that this cellular reprogramming occurred at that inflection point, but actually the reason there's a pink arrow there is because we actually found out it occurred right up front, even during this cytokine storm phase. So we did a study about a decade ago where we basically did, firstly, a meta-analysis in a whole bunch of patients from the literature. We did a small clinical study at St. Paul's, and the bottom line was that we were able to demonstrate what you see in the box there, that early sepsis is associated with a diagnostic immune non-responsiveness gene expression signature. And this demonstrates immune cell reprogramming or endotoxin tolerance, which is a kind of a form of cellular amnesia. So the cells just forget how to respond to an infection. And critically, it's actually mediated by altered macrophages in the patients. And this was, we originally demonstrated this, and it was actually subsequently proven by other individuals. So we performed a large prospective multi-center clinical study with more than 500 patients to confirm these data. And what we wanted to do was refine the signature that in 2014 was 31 genes. We wanted to correlate clinical symptoms with transcriptomics using machine learning approaches, and that's an identification of things called endotypes that I'll come to at the end. And we wanted to also extrapolate to additional patient populations. And we've shown that COVID-19, pancreatitis, perforated appendicitis, and pediatric sepsis are all different forms of sepsis. So what we did is we collected blood into Paxine tubes at countries all around the world. You can see the sites for Canada, Australia, Colombia, Netherlands, and the USA at first clinical presentation. So when the patient appeared in the emergency room. And then the blood was transported to Vancouver, where we did high throughput metagenomic sequencing and high-end bioinformatics. So this was kind of an AI-driven discovery methodology before AI became popular. So I wish I discovered this 5 years later and I would have been famous. But anyway, so it turns out that what we did is we had 572 patients. We did blood RNA-seq on that, which resulted in expression of 11,000 genes. We had a strong hypothesis, which is that certain genes were more important than others. And we used the method called LASSO, which is an AI methodology, to sort out which of the genes were most important. And this actually got us down to a set of 6 genes, and you can see the PCR progress plots over on the bottom right-hand corner. And you can see that we get beautiful PCR profiles for these 6 signature genes. And we compared this to 2 housekeeping genes that don't change according to whether a patient has sepsis or any other severe disability. And then basically we use another machine learning algorithm that is also AI-driven to actually give a diagnosis of whether a patient has sepsis or not. So just to show you that it actually works, these are some data. We have a lot more than this. So in the top slide, you can see basically a large number of patients in the ICU. This is in 2 cities, one of which happened to be Toronto. And sepsis COVID patients in Quebec, a bunch of emergency room patients in 4 different countries and 4 different continents throughout the world, and also a direct PCR test on patients in Toronto run together with Claudia dos Santos, who's from here. And what you can see is an AUC, which is an overall indicator of how well the test works for all patients. You can see the word sensitivity or sense, which means how often that test accurately diagnoses sepsis, and then accuracy, which is a balance between how well it diagnoses sepsis and how well it diagnoses non-sepsis. And you can see all of these numbers are pretty good. It's worse in the emergency room than, in other words, those very, very early patients. But within a few hours of the patient being in hospital, the test gets up to about 90% accurate. Recently, we have a company that we own a certain percentage of in China, and they've done a study test on 200 patients completely independently from us, different reagents, different operators, different hospitals, obviously different patient population, and they've come up with virtually the same data as you see in that sepsis PCR test in Toronto. So Toronto is the same as China, at least with regards to sepsis diagnosis. I told you there are a couple of other PCR signatures out there. One Septicide has already been released onto the market. It's not a very good test, unfortunately. And the other, Inflammatex, is just being resolved right now. It's a much larger gene expression signature and therefore can only be assessed by less accurate methods than polymerase chain reaction. And it's pretty okay, but not quite as good as our test. And then you also see there cancer-negative control. And so this is not diagnosing that patients are acutely unwell. It's diagnosing whether they have sepsis or not. And in addition to this, we've also done a set of patients with cardiogenic shock, a set of patients with severe inflammatory bowel disease, and none of these patients are predicted in severity by our PCR test. So remember, the gray is the status quo right now. What we would like to think is that we can now come in with a rapid diagnostic assay, and it's our proposal to enter formal regulatory clinical studies in the next couple of months. And that will now inform the physician of a certain probability of sepsis. We have accumulated nearly 3,000 patients worth of data right now. So we think it's got a really good chance of getting through regulatory studies, and will inform the physician that there is a 70 to 90% chance of that patient going on to sepsis, allowing immediate and patient-specific treatment regimens. And it's been shown that if you can get those therapies started early, you'll improve patient outcomes, have shorter ICU stays, and fewer deaths. And we've commissioned an economic study on this, and it looks like we should be able to save at least the US medical system a couple of $2 billion a year by instituting earlier diagnosis of sepsis. This is a very, very, very big deal. So it's a groundbreaking diagnostic technology which basically delivers results in approximately an hour, and it will improve sepsis survival rates. Okay, so I'd just like to introduce you to the concept of endotypes. And one of the problems that we've had with sepsis is it's an extremely heterogeneous disease. The causes, the patient conditions, the situation with individuals, the demographics, the genetics of the patients varies dramatically from patient to patient. And so it's been considered so complicated you can't really deal with this disease at all. It's more like a syndrome than a disease. So Patricia already introduced the concept of endotypes with the idea that you should be able to sort patients into different mechanistic underpinnings, groups with different mechanisms, and then you can start to deal with them independently. And it was our hypothesis going into this that basically these patients would have different sets of genes contributing to their immune dysfunction. So I told you about the AI-driven discovery of early sepsis biomarkers. So we were able to use that same set of patients because we had gene expression on the total patients to sort out 5 different endotypes. And what you can see at the bottom is a result of K-mediated consensus clustering that clustered the patients into 5 different groups, and then Lasso AI that actually finds signatures that define each of those groups. And so this is a gene set enrichment assay that you see at the bottom, where the red bars mean that there's an enrichment of that particular set of genes that define that particular endotype. There's about 200 genes that are differentially expressed only in each of the particular endotypes. And I think you can also see, if you look at the sort of black to grey bars, the third bar down represents severity of the disease, and the bottom one represents mortality. And I think you can see that basically a couple of these endotypes are much more severe than the other endotypes. So we're actually able to isolate now 2 gene expression signatures that will diagnose each of the endotypes, so 2 genes per endotype, 10 genes in all. And they do this with about 90% to 97% accuracy. So sepsis demonstrates heterogeneity. We were able to define these different endotypes. I mentioned there are 200 genes for each endotype, and these are the pathways that are associated with those genes. And the bottom line is the pathways are distinct for each of the sets of genes. And there are 2 of these endotypes that show very poor prognosis, and 3 of the endotypes show quite good prognosis. And this is actually independent of outcome at this stage. It's just sorting patients into mechanistic groupings. So once you have a mechanistic grouping, you can start to think about basically taking these the patients who fit into that mechanistic grouping. I apologize for that. I should tell you there's a bomb cyclone in Vancouver where I'm going this evening, and so that's why I had my phone on. So I really apologize for that. I usually turn my phone off when I'm talking. Anyway, so basically we can sort the patients into which of these endotypes there are, and now we can use drug development methodologies using another type of AI methodology that now tells us which genes, drugs are likely to target the genes that are specific for each endotype. So I'm not going to tell you all about that because I don't have time, but I'm just going to tell you that the NPS endotype that has nearly 50% mortality, that endotype can be targeted by dexamethasone, which is a known agent that treats sepsis, but with somewhat indifferent results. So it works in some patients, but not in other patients. And we believe the reason for this is it should be targeting a particular type of endotype. So this is a way of starting to rethink about how you're going to treat patients with sepsis. And then lastly, I just mentioned that sepsis endotypes can be identified in severe COVID-19 disease. You see that basically the endotypes sort out in these patients with COVID-19 disease from Quebec. There were 129 of these. Basically, the severity and mortality signatures were also dysregulated in these patients. So COVID sepsis looks like it's sort of similar to normal sepsis. So to confirm that, we basically looked at these patients with severe sepsis, basically. What you could see is that we could see a distinct set of gene expression pathways, so these are the reactome pathways and the hallmark pathways, that were expressed in patients who survived, where they had a persistent, they had a particular type of response, so the pathways were dysregulated, and then those pathways went back down to baseline by day 7. And in patients who died, those pathways persisted from day 1 to day 7 in those patients. So it looks like basically there are these persistent immune dysfunction genes that occur in patients who die. They never resolve their disease. If we now start to look at the unique COVID mechanisms, which are captured in day 1 in the ICU, you can see that there are a whole bunch of pathways that are especially adaptive immune pathways, and the top ones are largely interferon pathways, as you'd expect from a viral response. But when you look at day 7, there's actually no difference in gene expression pathways. So by the time you get 7 days into disease from severe COVID in hospitalized patients, there is absolutely no difference between patients with severe COVID and patients who are from the general population who largely have bacterial sepsis. So in conclusion, sepsis is very complicated, but can start to be addressed by systems immunology methods, which I described today. We have a review coming out in Frontiers in Science that I think you'll hear about because it's going to be well publicized. It's their flagship journal. I want to particularly mention the 2 guys who are most influential in this, which are Arjun Baghala over in the middle there, and Andy Ann, who's over on the right-hand side, both on the right, and then a bunch of people in my lab and a whole bunch of collaborators. And I thank you for your attention today.
Thank you, Bob. So are there any questions for Dr. Hancock?
Thank you, Dr. Hancock, for that very interesting talk. I would love to ask you about the subset ER diagnostic development that you mentioned. I'd love to hear your take on the practical implementation, right? Because this is an RNA-seq-based type of technology, which I imagine can only be run by very advanced laboratories.
Yeah, so we had to do the original study by RNA-seq because we wanted to look at the whole gene universe. But once we'd sorted out which markers were most important, then we could switch to PCR, which is a common methodology available in hospitals. So we managed to make fairly good breakthroughs well, not us, basically in collaboration with others, we've made fairly good breakthroughs in extracting RNA now from blood, which can be done in less than 10 minutes. And now then you can do a simple PCR analysis where you use different coloured markers for each, different coloured dyes to reveal each of the particular genes that are part of our expression signature. And you always compare this back to housekeeping genes, and that means you don't need internal or external controls. So the test is pretty simple. It requires the hospital to take an extra tube of blood at the time that you're doing your normal workup of a patient, which occurs a very short time after the patient ends up in the in the emergency room. And then that blood basically is processed separately to do a PCR reaction. And for us right now, it takes a total of an hour, although we have some preliminary point-of-care device data that suggests maybe half an hour will eventually be the time.
Well, that's fantastic. So can you run this instrument straight from blood, or does it require enrichment via blood culture?
Okay, so currently it has to be run in a central lab in a hospital system. Although you can collect samples and pool them, in fact, that's not really advisable because of the urgency of sepsis diagnostic for any given patient. So you'd like to do that test quickly. And I recognize that there's going to be some difficulties logistically for that. But you can also pool the samples and also test them whenever you have enough to make it economically worthwhile. But the point-of-care device now, there's now these machines that look about the size of a bread box, where you just feed blood in one end and get a result in a half an hour out the other end. And that that'll be right at the bedside.
Yeah, that's very exciting. Thank you.
Thank you.
This sounds like a very interesting lab test, but I'm thinking from a lab implementation perspective, even if you did have a point-of-care diagnostic system or close-to-care system, how are you going to choose your patients so that you're not testing almost everyone? I'm thinking more from a lab utilization perspective.
How are you going to do what?
How would you choose your patients such that you're not testing almost everyone? Because using the surviving sepsis mindset, you want to find everyone, and the diagnosis can be subtle. It mimics many other diseases. So what is your threshold to test people, and how do you avoid a situation where almost everyone is getting a test, which is a lab management nightmare?
Okay, so I would say putting my company hat on, I don't want to make that discrimination. I want every patient to get the test. However, putting on a practical hat, which is the hospitals don't want to have unneeded costs, the way we did it in our clinical study in the 4 different countries is we asked physicians to go based on sepsis 1 criteria, which means 2 or more what they call SIRS symptoms, which is the altered blood pressure, temperature, heart rate, this sort of thing, and also mentation, and suspicion of infection. And that we got, from that criteria, we got about 50% of the patients actually came down with a diagnosis of sepsis. Yeah.
Great talk. I would like to ask you a question about your immunological amnesia. You suggested that this is a phenotype that's predominantly found in macrophages. I'm wondering if you were able to discern whether or not those were the monocyte-derived macrophages or tissue-resident macrophages.
Sorry, I used the word macrophages, but in fact we got the same signature in monocytes, blood monocytes. So even in blood monocytes, before they get converted into macrophages. So we actually showed originally it was in macrophages because those are easily accessible cells, but we've since been able to demonstrate the signature is expressed in monocytes, blood monocytes.
So do you suppose that the signature would be expressed in the tissue-resident macrophages as well? The ones that seed the organs and during development?
I would anticipate they would be. Certainly in collaboration with Claudia dos Santos, we have seen some expression in nasal swabbings, for example, where I imagine that's where the tissue-derived macrophages would be. So I'm guessing that tissue macrophages will also be expressed, but ultimately it comes down to what samples you can actually use, and you would want to use a sample that is actually freely available to clinicians, and I think blood is the obvious one.
Thank you.
Good afternoon. You mentioned that you made some calculation on economics during your presentation, economy of the...
Economic analysis. Economic analysis.
Oh, economic analysis, yes.
The economic analysis.
Yeah. So
could you extrapolate a little bit on the model you use and what were the inclusion criteria or the data that you included at the beginning of the model?
Yeah, so it was a company, RTI Health Solutions. They utilize publicly accessible data in the US about a variety of different parameters to glean times to diagnosis, differential mortality, ICU entry, and outcome rates based on when the diagnosis was done and such like. And so they put this into a giant modeling system, and that's what they came out with.
Okay, thank you very much.
Okay, if there are no further questions, we wish you a safe journey back home to Vancouver. Dr. Hancock.
Thank you.
Thank you.